Good. Okay. Well, my name's Dr. Trevor Brown. Welcome to Neurocare Academy. And today we've got a special guest. We're really fortunate to have Dr. Tomas Ros with us from the University of Geneva. Hi, Thomas. How are you going?
Hello, everybody. Nice to be here. Thank you
So
for the invitation as well.
that's all right. No worries. And look, we're really looking forward to hearing what you've got to tell us about all your research and the application of neurofeedback and particularly the clinical application of it. So it's early morning there in Geneva, and it's about 7 p.m. here in Melbourne, Australia. So we'll get stuck into it. Thomas is going to answer some prepared questions for us. And look, one of those questions is, where were you born and where did you attend university? So we'll ask Thomas to tell us a little bit about himself initially. Thanks, Thomas. Go for it.
My pleasure. It's an interesting story I thought I might share because I didn't end up where I was born. This is a map of Europe. So I was born in Croatia, a small country in the eastern of Europe, in the capital Zagreb. This used to be Yugoslavia, as many people know, but then there was a war back in the 90s, and Yugoslavia split up after the fall of the communist regime. So at that time, we decided to emigrate and move to London in England. So it was a bit of a cultural shock, me going from when I was about 10 years old to a different school, boys only school in England, coming from Croatia. But that's where I got my education afterwards. And just a little picture of Zagreb, where my city of birth in Croatia. I used to ski on top of this mountain you can see behind. It's about 1,000 meters. And I used to do a lot of skiing, and then science became my passion. But at that time, it was quite a nice, idyllic place to live.
Fantastic. Thanks, Thomas. And look, I've been to Zagreb. It's a wonderful place, beautiful country, Croatia. So look, and then you obviously have moved around the world a little bit and are in Switzerland now. So can you tell us a little bit about your research now, and what subject and research laboratory did you complete your PhD?
Yeah, so basically when I moved to England, then I did my high school education there, and I did a physics degree at Imperial College as my bachelor's. And then I did a master's. I went to France for a year to do neuroscience because I felt like I wanted to study a little bit like human behavior, and I thought physics was a little bit too dry for me in terms of just equations. And I was always fascinated by this kind of humanities as well. So I couldn't choose. So I thought, okay, man, neuroscience kind of seemed like a nice balance, or psychology as well. So I moved to neuroscience as my master's, and then after that, I was looking for a PhD, and I couldn't find one in France at that time. And basically, I moved back to London, where my family was, and then I started searching for a PhD there. After about a year searching for a PhD, looking for kind of a subject that interested me, but also like a position that was vacant, I actually met John Griselli randomly at one of the conferences there, one of the seminars where he was talking about neurofeedback. And then we started chatting after the conference. I asked him, do you have any kind of research positions? And slowly, surely, then we sort of started working together. And then I ended up getting my, sort of starting my PhD in his lab. And just to show you the lab itself, here is the lab in Goldsmiths at the time where he was professor. I'm not sure if you can see it. Here it is. So this is in 2006. So I got my physics degree in about 2001, and after my master's and so on, then I started to do my PhD. And you can see here is John on the right. Obviously, John is well known for his pioneering neurofeedback research back in the sort of 2000 in the 90s. I'm just next to him, and then there's Trevor Thompson, who's part of the lab. There's also Mac Chen and Tony Stefford, who are also doing neurofeedback research. So yeah, this group is now split up into different sort of jobs. Some people are still doing research. John is also doing a little bit more, like he's an emeritus professor now. But then also there was the SANS Society that was founded at that time. And yeah, it's some nice memories of London where I started my neurofeedback research. And I just wanted to show, like, in the context of the history of neurofeedback, you could think this is like the full. You can see here how many publications per year in this domain. And let's say here's where I started my PhD around 2006. And you can see it was just before this kind of second wave, you can call it, of interest in neurofeedback. And interestingly, this first wave here was peppered with some very good publications, like in some famous journals like Science Journal by Camilla, for example, in 1978. That's kind of coincident with this first bump in interest in neurofeedback. And then there was also another paper in Science by Betty et al. In 1974, and one of the first papers in Science on the theme of neurofeedback, which involved like operant conditioning of animals and sleep spindles, was by Barry Sturman back in the 1970s. So you can see that even though you had this initial interest in neurofeedback and there was some kind of increase in, let's say, research output, it sort of fizzled out during sort of the 1980s and 90s. It became kind of went into hibernation mode and somehow non-linearly just came out again, which is quite a mystery of how this occurred. And it's a very interesting timeline. And then this is where I picked it up, with John, who was kind of holding the flame a little bit at the beginning of the 2000s in Europe, at least. And then it was increasing interest in neurofeedback, now even higher. And this was actually a picture we took with Barry Sturman back in 2006 at the first Society of Applied Neuroscience conference, which one of the themes was also neurofeedback. So this is one of the boys' club that around Barry, that were some neurofeedback researchers with me just here, and Martin Arns here on the right of me. And then basically, if you fast forward to the present, even though it's such a humble beginning, let's say, and it took a very long time for neurofeedback to come through, at least in terms of research, we published a paper in 2017 in Nature Neuroscience Reviews, which can be seen like a little bit like a litmus test about whether a field is being accepted by the scientific community and the medical community. So that was a really nice achievement for the authors that are also doing fMRI neurofeedback. And ultimately, we also managed to publish this consensus guidelines on neurofeedback research in 2020, based on a whole community of neurofeedback researchers. You can see here the full list. It's about 80 authors that have signed up for more rigorous and transparent neurofeedback research. So we think that the future of neurofeedback and the research domain is bright. And hopefully, there's much more to discover. Obviously, we could say this is just the beginning.
Fantastic. Yeah, look, obviously lots of research that's gone on over the last couple of decades. And, it's a really exciting time in neuromodulation, particularly in neurofeedback. You've done some research in fMRI, as you've said, also other neuromodulation techniques, TMS in particular, you use sometimes as well. So over the past 15 years, Thomas, I mean, I know a lot of your research. I cited you a bit in my own PhD. So that was, it was fantastic to finally meet you and now have the opportunity to speak with you. But what would you consider as, I guess, your top 3 research achievements and scientific publications that you've sort of achieved so far over the last 15 or 20 years?
Yeah, that's an interesting question, because there's a lot of research output that one does in a career. I think there's something like 30 or 40 publications so far, but a lot of them are collaborative. So it's not just down to me in terms of it's a whole group effort. But if I had to choose, I would choose some of the research that I did at the beginning of my career, where things were not so clear at the time in terms of neurofeedback, and some of the things that we did was working on top of those initial discoveries that we made, like choosing the protocol and choosing what kind of modality you want to use. So I'm just going to go through some of those papers using some slides. So the first paper I would like to share with you, the first study, is based on investigating neurofeedback with TMS. So this was sort of a seminal paper because nobody's done that before in terms of looking at whether you can detect changes in the brain due to neurofeedback, but with TMS. So not TMS as an intervention, but TMS as a kind of a probing tool for looking at changes in plasticity in the brain. It's a very sensitive tool for probing plasticity. That's why I decided to use this idea. And the other thing that was novel was that you wouldn't look at changes maybe pre to post after many sessions, but after just one session. So was it possible to even detect some kind of tangible changes in the brain only after one session of 30 minutes, which you would think, it's quite brief, and maybe there's something not even detectable there, or that maybe there were changes that would occur, after sleep or so on. But we went to directly test this hypothesis, and it was a collaboration with Queen Square at the Institute of Neurology in London. And you can see TMS, basically you stimulate a part of the motor cortex, and then you sort of record the muscle response, usually on the hand, and the sort of the strength of that muscle response, how strong is the twitch of the muscle, is essentially proportional to the synaptic pathway, i.e. how strong is the connectivity from the motor cortex to the muscle. And that's summarized by this slide, where you can see you stimulate the motor cortex here, and then you basically have this transynaptic pathway that then invokes this motor evoked potential, which is called the MEP. And the size of the peak of the MEP basically tells you how strong is the pathway. So the bigger the peak, basically the more excitability you have in the brain. And what we did was we basically then looked at if you apply a neurofeedback to the motor cortex, so to the EEG that's emanating from the motor cortex, and you try to activate that EEG. In this case, we try to downregulate alpha, which is a rhythm that's usually inversely related to cortical activation and metabolism, whether that would then increase the MEP post neurofeedback after about half an hour of neurofeedback. So that's what we tested compared to the baseline. And what we found was, firstly, that yes, this alpha amplitude in the motor cortex, you see here on the left hemisphere trained, at rest is quite high, and that basically the objective is to decrease it with neurofeedback, and indeed it does decrease over time, and each one of these training periods is 3 minutes. So you have 30 minutes in total, and you can see significantly reduced alpha during neurofeedback in both hemispheres, actually. And then how does that translate to the MEP change? So this is the mean MEP as a percentage of the time zero, the baseline. So at the beginning, at the end of that training, you can see that the MEP didn't change significantly immediately after. This is at around 5 minutes after the end of training. But as you wait longer, so here around 20 minutes, this is time 2, post neurofeedback, suddenly there's this quite a significant increase in excitabilities, around 25 to 30%. That basically also is proportional to the time elapsed. So in fact, excitability changes with time proportionally, and so it's unlikely that this is a kind of arousal effect, because the arousal effect you would have maximum at the beginning, and then it would go, it reduce with time after experiment. Here, it actually ramps up significantly with time. So that makes us think that maybe there's kind of molecular changes in the brain going on that take some time to ramp up, but that induces the plasticity in the brain long term. So that's one of the studies that, and if people are interested in knowing, well, how does this overlap with other treatments that modulate brain excitability? So TMS can also be used in repetitive fashion to, for example, also augment excitability. And here you can see the frequency at which you administer the TMS train basically determines whether this protocol is inhibitory, like in this case, 1 Hertz. You see here the baseline, the MEP decreases with 1 Hertz TMS. But as you increase the frequency, you kind of excite the cells further, much more quickly, and they actually then turn into more excitable neural cells. And you can see here with 20 Hertz TMS, it's about 30% increase in excitability. And with neurofeedback, I overlap this with the error bars and the effect size that we had from this experiment. And you can see that there it sort of overlaps with TMS in terms of effect size. So that's a very interesting finding, because it would mean that maybe even using a natural method like neurofeedback, which doesn't involve external fields, you can still get the maximum out of that neural substrate in terms of excitability. And if you compare in terms of how long it takes, well, TMS obviously has an edge, because if you stimulate at 20 Hertz, you stimulate only for 1.6 seconds, and you get that excitability increase. With your neurofeedback, you have to basically do it for 30 minutes. But on the other hand, it might be safer. So that's something to be investigated still. And then if you look at, what are the actual clinical or mechanistic sort of applications of this, is that, traditionally we used medication to change excitability of the system. And in some grand scheme of things, there's the uppers and the downers, or the stimulants or the depressants. So you can consider the stimulants being like kind of like what we call amphetamines, like methylphenidates, which actually have been found to increase excitability. Here you can see there the MEP goes up, and these are so-called dopaminergic agonists or noradrenaline agonists. So they kind of usually increase the sort of metabolic activity of the brain and speed up cognition, and sort of they're usually used to improve attention and so on. On the other hand, if somebody's overexcited or overactivated, and thoughts are racing and so on, then usually you use diazepam or some kind of tranquilizers to decrease excitability of the brain that make them allow them to fall asleep, for example, like GABAergic agonists, and they usually decrease the MEP. So in this case, you could replace some of these TMS protocols with this alpha, decrease to increase the excitability, or alpha increase to maybe decrease the excitability. So you might find another way to simulate the effects or emulate the effects of the drugs, but using a natural training method. Yeah. And then the second study is taking a bit longer. Second study is with alpha, that we do the same protocol, but we looked at whether that has an influence on behavioral performance. And in this case, we looked at mind wandering. So mind wandering being defined as changes in thoughts that are not relevant to the task. And in this case, some studies, previous studies have shown that alpha is kind of used as a gate to block sensory information. So it can be used as a proxy to assess whether the brain is internally oriented, in an internally oriented state or an externally oriented state. So for example, when alpha is low, the gate is open, and therefore you're receiving visual information or information from the outside. That's usually when the brain quietens. And conversely, when the alpha is high, usually you're blocking sensory information, and that you're kind of retrieving internal information like thoughts or plans and so on. So the brain is constantly swapping in and out of this kind of state of internal versus external attention. And this is correlated to the alpha, as you can see in this part here. So when alpha is high, you're in an internal state. That's when you're kind of mind wandering, thinking. Alpha is low, you're eternally attentive, and there the alpha is low, and your thoughts are lower in this case. And so you have this kind of fluctuation. The question is, if you were to train to downregulate alpha, for example, to increase this state of externally oriented state, would you have a decrease in thoughts? So that's what we wanted to test. And we use the same kind of protocol, one session neurofeedback, where you can see here now we had a sham group, and basically the neurofeedback group was able to successfully decrease alpha, and the sham group, over time, the alpha went up. So there was this difference between these two groups in terms of entrained EEG. And what we found was that the neurofeedback group what was in fact correlated with mind wandering. So the alpha levels, those subjects that decreased alpha the most post neurofeedback, were also the ones who had a decrease in mind wandering. So we kind of confirmed this mechanism that we can maybe quieten the mind using alpha downregulation. So this would have implications also for other brain disorders that have too many thoughts, like depression or schizophrenia and so on. So that was a nice finding. And also we found that this decrease in alpha was linked to an increase in a special network in the brain that's called the cognitive control network, the salience network. And here we found that neurofeedback led to some kind of metabolic increase in the frontal cortex that we didn't find with sham. So again, there were some kind of downstream effects in the brain that you could detect that was linked to the behavior. So again, some kind of triangular evidence that you can impact the brain with neurofeedback, and that this is linked to cognitive control networks and also changes in behavior. And lastly, this is a more recent study, although it's something that we didn't expect based on the neurofeedback model. And this was where we applied this alpha downregulation protocol to patients with PTSD. And as I showed you, usually you have this high alpha at the baseline, which is here, and then you do neurofeedback, and then alpha reduces nicely. But what we found that in this patients particularly, and not in controls, there was an increase in alpha post neurofeedback, which was kind of paradoxical, because you would either expect it to be the same as the baseline or lower, because you're going in the direction of training. But in this case, these patients had an almost opposite effect, which was very confusing for us at the beginning of why was this happening, because we wanted to kind of induce lower alpha. But what we found out later with another study when we recruited a control group was that in fact, compared to controls, as you can see here, the mean at the baseline is actually lower than controls. So here T1 before neurofeedback. So they're kind of, let's say, pathologically lower in alpha or abnormally low in alpha. So that maybe explain also why their symptoms are hyperactive in terms of arousal and so on. And that interestingly with neurofeedback, there's a renormalization of alpha, although you're going in the opposite direction. So we try to understand how this might work and why it occurs in that way. And some of the models indicate that it could be due to the homeostatic mechanism of the brain, which means that once you push the brain too far in one direction, it will kind of flip back to correct itself in terms of the excitability inhibition balance. So although you're exciting the brain and sort of going even further to lower alpha, then after the brain tries to recompensate, and then in fact you end up with a kind of therapeutic response that is more closer to the normative population. And the reason why I mentioned that is because then we looked at the correlation between the changes in arousal and the changes in alpha. And in fact, those patients that had the most alpha increase post neurofeedback actually had the greatest decreases of arousal. So this could be therapeutic for PTSD. And my collaborations with Professor Lanius in Canada, there's a few studies that we looked into, and some new evidence from the randomized controlled trials seem to indicate that this is a good protocol for PTSD. So we're going to be looking into this more deeply. But it's an interesting paradox that you don't sometimes end up with the hypothesis you expected, but that still there's some positive things that you learn about mechanisms.
Yeah, look, that's great, Thomas. Thanks very much for that, and really, it's some instrumental work that you've done over the past decade. And those studies that you showed us were amazing. If that been an inspiration to a lot of people I know in the universities around Australia and around the world. So the research you're doing is fantastic, and your import into the community is great. And I noticed there's a real clinical benefit there eventually, and your focus is on making this clinically applicable. So that's what really interests us here at Neurocare Group as well, because obviously with the clinics around and trying to actually assist people through the neuromodulation techniques. This research takes a long time. There's a lot that goes into it all, and the fact that it's clinically applicable at the outcome is great, and you obviously have that focus. So that's the research that you've done. You've introduced to us today. What about the current focus of your work at the University of Geneva at the moment?
Sure. So basically, it's still in the domain of neurofeedback. I kind of dedicated my research career to this area because I don't think it's explored enough. So we continue on that theme, and sometimes it's been difficult to continue because you apply for some grants and they don't get funded, and then usually the way to do it is like to change the topic or change the domain because you're not successful. But there's been a bumpy road for me academically to stay where I am in academia. But I will show you where I am, and luckily there's a group here in Geneva that is specialized in EEG, and recently they started to take an interest in neurofeedback as a method for neuromodulation. So then I came into the picture, and this is the group of Professor Michel, who works also with EEG and microstates. So I'll just explain a little bit what microstates are. So actually just a little slide to show you what the building where I work at the moment. This is an interdisciplinary center of different universities in sort of the Geneva area, including the University of Geneva, EPFL, the École Fédérale Polytechnique, and this sort of center of biomedical imaging, where I'm also a senior staff member. Basically, we look into how to bridge the gap between, basic neuroscience and EEG and clinical translation. And so this is in this campus biotech, where we collaborate, and essentially we're collaborating on this topic called EEG microstates, which is a slightly different perspective on the EEG. Usually you look at EEG on a sort of electrode level, and you calculate power and spectra and so on. So here the idea is to look more at the spatial configuration of the fields themselves. So you kind of take into account the whole spatial distribution of the EEG power, and how these kind of spatial patterns might repeat themselves over time. So basically, what are EEG microstates? I'll give you an example. If you were to take just one slice, just one sample time point, in a particular time, you have a field, right? And so, for example, you take these are seconds here, and if you take around 0.5 seconds, you take exactly this one red line, you will have this kind of field configuration, which is sort of positive in the front right prefrontal cortex, and then less positive on at the back. This is so-called a microstate A map. Then if you take a different time point randomly, you might come across this other configuration, which is exactly the same but symmetrically kind of mirror. This is a microstate B map. And then there's also a few other maps that are common, like this one, the frontal sort of occipital one, and then another one that looks more like positive around the frontal electrodes. So the brain is kind of going through into these different configurations. They're basically different EEG generators that generate these maps, or let's say networks. And the interesting thing is that if you look at how long each of these maps sort of maintain themselves, like the sort of stability of the map, is that in fact they do repeat, and they are not just stable in one sample time point, let's say 1 millisecond, but they last longer. And so if you then would cluster these maps in terms of how similar they are, and using sort of clustering algorithms, you can find out that in fact they occur in kind of trains, and they're stable for at least around 80 milliseconds or so. So for example, this map, the green map A here, you can see there's a period of time that it's stable for, and then it switches to the purple map, which is map D, and so on. And they're flipping in and out of each other. Sometimes they're short and sometimes they're longer. And it's the distribution of the states that kind of people have found that are related to some behavioral correlates. And so you can consider them a little bit like attractors in the system, or preferred network states. And how long these states are prevalent for seems to impact behavior in different populations. For example, here, this is a recent paper published in Nature Communications that shows that microstate D, this is the class D map here. You see, most people, even healthy controls and patients, have this map. So it's not like the configuration is wrong. The configuration exists almost similarly across different populations. So these maps are kind of conserved, but it's the temporal aspect of the maps that seems to be abnormal. So for example, here you can see that the mean duration of this microstate of class D here, you see in siblings and controls, controls have a long class D, a longer class D than patients and siblings of the patients. So in fact, this microstate is shorter in duration, and that might lead to the cognitive impairment that you see in people with schizophrenia, for example. And the same thing in terms of time coverage. You see the time coverage is longer for class D, again, in healthy controls. So the duration is shorter, plus the state is not visited enough. So one idea would be to try to change this temporal activity using neurofeedback. So basically detect online these maps, how they occur, calculate in real time, okay, is the map present or not, and then ask the patient to try to, for example, in schizophrenia, to increase the prevalence of this map. In fact, so that the attractor becomes more imprinted in the system and is visited more often. And through plasticity, then, therefore, to reinforce that that attractor, and then restore the imbalance, and then hopefully see some changes in cognition. So that's the idea that we're using at the moment. And there's a few sort of hypotheses we have. We're looking at this for ADHD, for example, whether there's some differences in ADHD in these attractors and topographies. So this, you can use this generalized kind of method over many different disorders, and hopefully we find something interesting there.
Fantastic. Yeah, and we can see the clinical application straight away. And obviously these very prevalent disorders like schizophrenia and ADHD, like you mentioned as well. So look, there's obviously a real world application of that sort of thing straight away. Obviously neurofeedback takes some time and a lot of repetition to create this neuroplasticity, but to be able to reproduce these microstates for longer periods of time obviously induces this neuroplasticity. So look, really, really important work. Thanks, Thomas. Also, what are your future research aspirations? Obviously, we've gone from past to current. What about the future? What are you looking to for the rest of your career, or in the immediate future?
Yeah, you never know. I mean, there's a few ideas that I have that I'd like to explore. For example, just a recent paper we published was based on some looking at whether neurofeedback is associated with neuromodulator release, like dopamine and so on. At the moment, this paper, we didn't find any evidence for that. So, you have to be prepared to fail as well, or maybe try again, but a different neuromodulator. And basically triangulate neurofeedback from many different perspectives. So use these different modalities. That's been my kind of goal, to sort of use different methods to look at neurofeedback from different angles and then get a bigger picture of what it's doing. So using fMRI, using TMS, using PET, and then of course understanding better the EEG, and mining the EEG patterns, because that's the best what we're working with. There's some limitations of EEG, like the low spatial resolution. On the other hand, the reason why I prefer EEG is because it has a good temporal resolution. So you can implement this neurofeedback loop very quickly, and it's very translational, which means that it's accessible. You can have it in clinics, you can use it at home, even with these kind of homemade devices or commercial devices. So I see more of a future in EEG rather than fMRI or so on, because it's more easy to deploy. So therefore, we have to live with the limitations that EEG has, but try to harness the strengths that it has. And the hard part is really to find these biomarkers, if there are any biomarkers that are specific to some kind of disorders or not, and so on. So that's going to be the hard part, and that's why at the moment we're both working in parallel with discovering biomarkers as well as implementing neurofeedback, rather than just going ahead and testing neurofeedback as a concept. I think has been proved, but we need to find this coherence between the specificity of biomarker and then use this experimental time that is quite long to do a study. So we need to be sure that that we want to invest into doing a study or randomized clinical control trial. So the preparation phase does take long, and it means that also neurofeedback isn't being translated quickly, but that aspect is it goes hand in hand with the translational aspect. And that aspect maybe is even more challenging. But as a sort of a jokey aside, what I would like in my future is that I get more grant funding, so that we can actually carry out the neurofeedback research, because that's been the main limitation, I think, for many research groups around the world who have started neurofeedback and then didn't get funded, or students, the new generation of students who would like to carry out neurofeedback research. From my experience, it's been a very tough time at getting funding. So I don't think I've had one grant under my name so far, unfortunately, from a sort of a serious sort of scientific institution, national or otherwise, simply because neurofeedback was still considered as a fringe movement at the time. And even now, when I apply, the conditions are tough for early career researchers to kind of break new ground with novel ideas. So usually, when I got funding, it's always on the coattails of an established professor or person established in another field that has said, okay, well, let's go in this direction. But what would be nice to see is that the scientific funding bodies actually consider this as a real method that should be investigated more deeply, because there's still not enough neurofeedback research, although it's increasing. But compared to like TMS and so on, it's a drop in the water. Right?
Yeah. Look, and we see the clinical application of it all the time, and the effects of it. And as you showed previously, the effects at a cortical level can be as much as those of exogenous neuromodulation techniques like TMS, but it's happening endogenously. So look, it's there's obviously a lot of work that goes into that, and academically, as an academic yourself, to try to continue to conduct that research and get the funding is a big challenge. And look, wish you all the best for all of that, because the research that you're doing is so important for us to then translate that clinically and use that clinically. So it's just such an important thing for us. So
thank you.
So if we come back to I guess neurofeedback in comparison to other neuromodulation techniques, do you think there are any advantages of neurofeedback in the clinical practice compared to, say, TMS or TDCS, for example?
So basically, I think that the potential advantages of neurofeedback lie in these three or four domains that I have on the slide, which are the safety, the efficacy, the cost, and the stability of the effects. Starting with safety, I think Neurofeedback is unique because it really is a natural training method. Although you are using a computer, it's a little bit like a cognitive training. So the computer is guiding you, but it's you, the brain is doing all the work, and it's you doing all the work. So basically, conditions are kept as normal and physiological as possible because you're not pushing the brain into some kind of other areas where it's not comfortable or it doesn't usually operate, whether it's external magnetic fields or currents and so on. So and this also shows you that with these homeostatic mechanisms, that's why maybe the brain does change its tune after a certain amount of training, because it will detect that it doesn't like to go there, so it will engage new mechanisms to row back those changes, or flip them, or so on. So in some ways, you're letting the brain continue being in its comfort zone, right? And then, as we know, also with muscles, like once you get tired, you might be doing some gym training, you get tired. Okay, well, now I'm tired. That's a signal for you to stop. So you're pushing your brain or your body as much as possible, but not further than it is comfortable with. So I think safety is a very big deal, especially with brain health, especially when we're dealing with children and people who are developing. And we also know there's very few adverse effects on neurofeedback. I mean, there's been randomized controlled trials now done with hundreds of children and control populations, and the main sort of adverse effects were maybe been headaches and so on. But I think the adverse effects are much smaller than, for example, medication and so on. So there, I think, is a big plus. Now, in terms of efficacy, I already showed a slide on that. So I think you can probably attain changes that are on the same level. On the other hand, it does take longer, whether it's within session or across sessions. So you might need to do 30 or 50 sessions. But if neurofeedback is implemented maybe at home, then this can be managed if they do it like on a daily basis or so on, half an hour or so on. So I don't think that's a problem. In terms of cost, yeah, these EEG devices are becoming cheaper and cheaper. You've seen some of these epoch emotive things that you put in your head, that you can use to like play video games and so on. So maybe we can leverage all those technologies that are sort of consumer grade devices that also could perform neurofeedback in the future. Although for those we would need maybe more electrode coverage, so they might get more expensive. But on the other hand, if there's more competitors in the market, things become cheaper. So it could be that you could get some of these devices, a few $100s. And the clinics could lease out to people or have them and have many of them. And because essentially neurofeedback is just a sort of software trick, right? It's just a closed loop that the computers analyze, and with increasing computing power, these things should be like a cinch for future generations to implement. And essentially also, and the last point, which I think is the most interesting, is the stability. So whether these effects actually maintain themselves and whether they last, which we know with other therapies, you administer a drug, then things are great, and then as soon as you want to stop the drug, well, things go back to normal, because in some ways maybe it's possible that the brain is adapted to that drug and now has become dependent on it. So this is a completely different model of how you would try to treat brain problems, in terms of the paradigm shift, in terms of doing work from the inside versus actually imposing something from the outside. So that's why I call it like training from within, because it's the system doing the training. And it's a little bit like if you take steroids to improve your muscle strength, yeah, you can do it, but then as soon as you stop the steroids, the muscles just go back to normal. But if you actually do the work yourself, then that sort of increase in power that you have in your body is sort of built in to the system, and it's robust. And once you stop, there's a very long time until which it will decrease. So it's possible that with neurofeedback, and we've seen evidence for this, in multiple papers where they do follow-ups at 12 months and even a few years after the training, with no intervening training, and the effects are actually conserved, which is not the case for other treatments, from what I know. So that could be really a different way to look at things and do things. So, and then you could even consider thinking about calling it a cure, because after that, you don't really necessarily need to do more. But it depends on, obviously, if the disorder is degenerative or not. So for non-degenerative disorders, maybe you can reach a new configuration where the brain then is independent and doesn't need further tuning and is happy with the state it's in, and then it just continues in that state for the future. But of course, for other problems like multiple sclerosis, Parkinson's, these things, they could be like top-up training sessions, but they will never really change the disease from what we understand the mechanism. So for that, of course, we need different approaches like genetic interventions and so on that might cure the underlying cause. But for some kind of brain disorders that are linked to the brain configuration, the synaptic connectivity that is subtly different from the norm, there, you can imagine that you could tune it to a certain way. And then if the system doesn't deteriorate over time, then it would be stable technically after neurofeedback.
Great, thanks, Thomas. Look, you may have already answered some of this already, but this last question for you: what do you see the potential clinical translation of neurofeedback might be in approximately 10 years' time? Obviously, you talked about a lot of the advantages of it already, but how do you think that might change in the future?
Yeah, so I really hope that it will have some clinical translation. At the moment, it's very weak. If you look at hospitals, yes, there are some clinics that do it, but this is not sort of mainstream way of doing things. And I think that's what's missing. I think the public needs to know a little bit more about their options when they go into treating some disorders, and we need the evidence base to back that up. So yes, the evidence base is growing, but we need much more, a bigger, critical mass to convert this into kind of policy changes where you would get this kind of treatment in the hospitals or other clinics. And for that, yes, we need to do the science. And for the science to be done, we need the funding. And the funding actually attracts more scientists. So you can see new generations coming, and they have new ideas, and they want to forget the old paradigms. So I think it's important that, as I said, like there is increased funding for research. I think we've broken the sort of glass ceiling for these journals to accept neurofeedback research, like Nature and so on. So I think there the work has been done. It's just a question of having more output and then possibly looking at, okay, where does it work well and where does it not work well, and not overpromise. So maybe ADHD is a good place to start. Then of course we can try to expand the focus to maybe other psychiatric disorders, even neurological disorders like schizophrenia, psychiatric disorders, or depression, and look at the specificity and grapple with this heterogeneity that we find in the brain that all treatments, I think, are grappling with. And then maybe even put different treatments side by side and seeing which one is good in this context. And also I would like to stress that neurofeedback is a way to tune sort of, let's say, the hardware of the brain, but we also need to consider that the brain is not just sort of the person, or the mental disorder is not just the brain in a vat, but it's also in a society, in social relations between people, in how you live your daily life. So I think I see neurofeedback as part of the arsenal to help deal with brain disorders, but not the only one, and not to over medicalize brain disorders. And here I'm talking more about psychiatric disorders, which we know are multifactorial. Maybe some neurological disorders have very clear genetic or otherwise sort of causes, and there neurofeedback might play a very good role, or the only role. But if you're getting into more complex territory like psychiatric disorders, we mustn't forget also that psychologists and psychotherapists and let's say the software of the brain, the programs that the brain is running, you're not going to be able to change that simply by changing some kind of wave. You can change the way, changing the wave will change the sort of excitability, sort of the comfort zone of the brain, so you might be able to use neurofeedback to help a person understand you when you're giving them psychotherapy, or calm them down. So it's a adjunctive tool to help rehabilitate the brain where it's missing some kind of activity or balance. But on the other hand, that needs to be joined with other treatments together holistically, I would say, to help the person as a whole, because the person is not just their brain isolated from the rest of the world. And I think some of the research from Ruth Lanis' lab also show that these adjunctive approaches are important. So in that case, they have combined psychotherapy with neurofeedback. So I can see the future of neurofeedback being in having a role, but not the exclusive role for brain disorders.
Yeah, so in 10 years' time, as you say, maybe we will understand a little bit more about how neurofeedback can be integrated into a full package of treatment and be used at the right time and for the right purposes. So yeah, look, that's really the goal for us, isn't it? To put it into our toolbox, our therapy toolbox, and understand when it is useful. So perhaps in the future we'll know a lot more about that and be able to use it more effectively. And it's already being used effectively, but that can always be improved upon and used in other areas. But as you say, it's not it can't treat everything. At the moment, the evidence is accumulating for ADHD and sleep disorders, and it's starting to get there for as you're working on the PTSD area in particular. But yeah, we need to accumulate that evidence and understand more about how we can use it. So look, that's going to be a really exciting next 10 years of to follow your research and to see how you can inform us about all of that. So thanks so much for spending the time with us today. And maybe you'll join us again in a year or two and give us an update on how things are progressing.
Sure. And thank you so much for the interview. It's my pleasure to take part. And maybe in a couple of years, there are not going to be such massive changes. Things are slow in science. It takes maybe a year or two to even publish a paper these days. But maybe in 10 years, there will be something that will change. And I hope I will continue doing this work because I love it. And I hope the funding will be there to, continue my career in this domain. And then we'll see what happens. And I hope also that there will be many more scientists around the world doing neurofeedback research.
Thanks very much, and we'll follow it closely. Thanks, Thomas.
My pleasure. Take care.